Hello, everybody, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled, A Novel Approach to Epigenetic Methylation Analysis. The study of epigenetics has emerged as one of the most exciting fields in the life sciences, and interest in the topic grows greater each day. DNA methylation, an essential epigenetic process, plays a key role in regulating gene expression, resulting in both normal and disease phenotypes. Today's webinar has been designed to illustrate a promising technique that could rapidly advance epigenetics research. Reduced representation bisulfite sequencing, or RRBS, can allow epigeneticists to obtain quantitative DNA methylation information across the human genome at a substantially reduced sequencing cost. So let's meet the panelists who will be discussing RRBS. Dr. Steve Kane is Director of Product Marketing at NewGen Technologies, and Dr. Ben Schroeder is a Senior Scientist at the company. Drs. Kane and Schroeder are going to describe and present data from a novel approach to RRBS. After the panelists make their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the left side of your console and then hit Submit. The panel will try to answer as many questions as possible. I'm John Sterling, Editor-in-Chief of Gen, and I'm going to serve as moderator. So if everyone is ready, let's get going. Dr. Kane will be our leadoff speaker. He will then introduce his colleague, Dr. Schroeder, who will continue with the presentation. Steve, you're on.
Thank you very much, John. So this is Steve Kane, and I am going to be introducing you to the topic of epigenetics, primarily with a focus on DNA methylation. And then I will turn it over to Ben Schroeder, as John mentioned, who will speak in detail around a specific sample preparation method that's been developed at NewGen, which is related to a technique called RRBS, or reduced representation bisulfite sequencing. So to begin with, by way of introduction, the field of epigenetics is very broad and refers to a modification that can take place within chromatin, typically that alters the level of gene expression within cells or tissues. So these modifications are independent of specific changes in the DNA sequence, but rather take place based on modifications to both the protein as well as modifications to bases within DNA. So we only have one genome. Species, in addition to humans, have one genome, but our epigenome can vary with several phenotypic factors, such as our age, the tissue that's being studied, environmental factors, as well as different disease states. So really the phenotypic expression in a cell or an organism relates to both the underlying DNA sequence as well as the epigenetic modifications that have taken place within the chromatin. So these changes with respect to the chromatin proteins take on multiple different factors. There's methylation changes at different amino acids, phosphorylation changes, acetylation, as well as ubiquitination. At the level of DNA modifications falling under the epigenetic category, DNA methylation is really the one that's been studied the most deeply. And these modifications take place at cytosines, which we'll detail throughout the presentation. So it's specifically with respect to DNA methylation. There's really been a lot of research conducted over the last decade and beyond into the broader topic of methylome research because of the broad impact it has on a range of different biological processes as well as different specific disease states. So altered DNA methylation has been linked to gene expression changes in a range of different model systems and diseases. For example, in the area of cancer, there's both hypomethylation, so reduced methylation at specific sites, as well as hypermethylation that can relate to various aspects of cancer. For example, the formation of tumors, response to chemotherapy, relapse from therapy, a range of different categories where DNA methylation changes are important in the area of cancer and cancer research. Also in developmental biology, so embryonic development and stem cell phenotypic expression around drug and alcohol addiction. interestingly enough, can be related to epigenetic changes, as well as psychiatric disorders, healthy aging as it impacts the brain, and then environmental factors that relate to changes in identical twins. And I'll show you one example of that in a subsequent slide. From the pharmaceutical point of view, in particular with respect to cancer, it's an important area in translational research and therapeutic development. And in fact, several DNA methylase inhibitors, so inhibitors that would inhibit enzymes related to the DNA methylation process, are in use for the treatment of cancer. So here's a very simplified model of how altered DNA methylation might relate to changes in phenotype. And generally speaking, a change in the methylation state of upstream elements related to genes, so on the 5' side of promoter elements that regulate gene expression, altered methylation can in turn lead to downstream changes in the expression of different genes. So you'll notice on the left portion of these two figures a term CPG islands. This is a term that Ben will elaborate on in his portion of the talk. But a CPG island is generally defined as approximately a 200 base pair sequence that has a high level of GC content. And in particular, this GPG dinucleotide, which is the target for methylation changes in higher species. So typically when these CPG island sequences are undermethylated, you have a enhanced level of gene expression. And when they're highly methylated, or in a relative sense, more highly methylated than a normal gene, for example, in cancer, that can lead to gene expression being repressed. So a good example of that in the area of cancer is hypermethylation of promoter elements in different tumor promoter genes. That leads to a silencing of those tumor suppressor genes, thereby leading to a repressed expression. So here's another highly visual example that I wanted to share just to show you the very interesting fact that these methylation changes are inheritable. So you're looking at an example of how genome-wide analysis of DNA methylation has shown that various genes retain parental levels of methylation in gene promoter regions. So in this experiment, what you're looking at on the lower right portion of the figure is two different mice that are genetically identical, so they have the same age and sex and the same DNA sequence. But in one case, the mother of one mouse received a normal diet, whereas the other received a diet that was supplemented with methyl donors, which would drive higher levels of methylation on the DNA. And you can see this obvious phenotype difference, the mouse on the left having a much larger body weight. So this was inherited even though the induction of these epigenetic changes took place in the mother. So there are a number of examples where DNA methylation changes can be inherited by virtue of impacting various promoter elements, and then in turn gene expression changes that can impact the phenotype. So back to the area of cancer, this is just meant to show you that the amount of research that's been done and the studies of DNA methylation related to cancer are continuing to rise dramatically, beginning with this paper published back in 1983 by the Vogelstein groups. This is over 30 years ago. They observed general hypomethylation in various genes in cancers related to their normal counterparts in normal tissue. And since that time, you can see a dramatic rise in the number of publications that cite DNA methylation related to cancer. In part, this is driven by the advent of next-generation sequencing several years ago. And certainly the amount of people that are studying methylation as it relates both to cancer phenotypes as well as gene expression changes in cancer continues to rise. So I wanted to introduce you to some very general approaches to how people will study altered DNA and DNA methylation, and then we'll focus in on one specific technology that Nugent's developed. So there's three general approaches. One is an enzymatic partitioning strategy. So in this approach, during sample preparation to study DNA methylation, researchers would use methylation-specific enzymes to cut the DNA restrict the DNA and thereby separate it into methylated and unmethylated bins for further analysis. There's also different capture approaches using either a chromosome immunoprecipitation approach or a methylation DNA precipitation approach, or MeDIP. Here what you're doing is you're using an antibody or a binding protein to grab onto these CpG methylation sites. So it's an affinity purification method to selectively pull out methylated genomic fragments. This method has a disadvantage of really emphasizing highly methylated regions, very dense regions of altered methylation, and doesn't provide information around more sparsely methylated sequences. And then lastly, bisulfite conversion is a means of chemically modifying DNA such that you can study specific methylation sites at single cytosine residues. So this is really the only approach that allows you to achieve base pair resolution of cytosines versus methyl cytosines. And so the RRBS method that Ben is going to elaborate on is really a way of combining this enzymatic partitioning approach together with bisulfite conversion reactions. And then lastly, in my section of the webinar, I wanted to give you a brief overview of the two different products that Nugen has released for studying altered DNA methylation. And it's highlighted here with some of the key features of these different sample preparation methods. Each of these allow you to prepare libraries for next-generation sequencing. So all the reagents that are required for conversion of DNA through to a library that can be used for sequencing are provided in the reagent kits that enable these two different technologies. On the left is the Ovation ultra-low Methyl-Seq system, and on the right is the topic of today's webinar, the Ovation RRBS Methyl-Seq system. So in terms of comparing these two different approaches, the target region that's addressed with the ultra-low kit is a whole genome. So this would be a whole genome bisulfite sequencing method, whereas the Ovation RRBS Methyl-Seq system enriches for CpG island-containing sequences in the genome, so hence the term reduced representation. The basis for detection of methylated cytosines in each case is bisulfite conversion. However, in the case of RRBS, we've combined this approach with a specific restriction enzyme digestion termed MSP1, which Ben will elaborate on in quite a bit of detail. The source of the DNA with respect to the ultra-low kit is very flexible, so a range of different species, human and others. In the case of RRBS, it's also flexible. The commercial kit is quite suitable for human, mouse, rat, and other higher vertebrates, but we would recommend that the genome be analyzed for MSP1 content because there is specific restriction enzyme digest as part of the workflow. The input amounts of DNA required to carry out each process is low in both cases. The ultra-low kit uses a minimum of 10 nanograms of genomic DNA, whereas the RRBS kit requires 100 nanograms. If you look at the relative sequencing costs and the required depth of sequencing with each of these approaches, the Ovation ultra-low method is a whole genome sequencing process. You're really doing, in the case of human, a whole human genome sequence that has been bisulfite converted. So the cost is going to be higher in a relative sense to the RRBS method, which can have as much as a 50-fold lower amount of sequencing, and as Ben will show, to obtain largely the same amount of information. So that's really one of the key benefits of this approach. With RRBS, you're getting a large portion of the same information that you would gather from a whole genome sequencing experiment with dramatically reduced cost of sequencing. And then a couple of nuts and bolts about each kit. These are library construction methods. So with respect to multiplexing the libraries during sequencing, we offer barcoding options up to 96-plex that can go higher with customized solutions. So that allows you to, again, have benefits of cost reduction by virtue of doing multiplexing during the sequencing process. And then the overall workflow of each process, starting with genomic DNA through the construction of a library that's ready for sequencing, is a single day in each case. So there's a highlight of the features of each product. Now I'm going to turn it over to Ben, who will elaborate on the RRBS method and provide some details around how that kit can be used and some data that we've generated using this method. Turning it over to you, Ben.
Thank you, Steve. I wanted to start with a brief history of RRBS. This technique was first described in 2005 by Alex Meisner et al. In 2010, they continued to improve the method, switching from earlier restriction enzymes to MSP1. MSP1 recognizes a 4-base restriction site, CCGG. And as you'll notice, the central CG is the methylation motif in mammalian genome. So this switch to MSP1 was a key component of targeting the RRBS approach to CPG-rich portions of the genome. In 2012, further improvements were made in terms of removing a gel isolation step and phenol chloroform extraction steps, making the process much easier to complete. We've continued to improve upon the method, and I'll describe in more detail the current state of the art. Before I do that, I wanted to show you this table from Gu et al., which describes several different approaches to analyzing DNA methylation. And in particular, I want to draw your attention to the columns marked RRBS and whole genome bisulfite sequencing, or WGBS. Circled in red are the percent of the genome covered by each technique, 9% for RRBS and 76% for whole genome. And below that, the number of Illumina reads per sample required to analyze that fraction of the genome. For RRBS, you can see approximately 10 million reads required, and for whole genome, greater than 500 million reads. So to go from 9% of the genome covered to 76% of the genome, to get that extra 8x coverage, requires more than a 50x increase in sequencing. So the approach of RRBS is actually quite efficient in terms of its sequencing requirement. The other technique involved in RRBS is bisulfite conversion, and I wanted to spend just a brief moment to explain how this chemical process works. Cytosine can be reacted with bisulfite in the process shown at the top of the slide, ultimately resulting in its conversion to uracil. However, 5-methylcytosine, shown below, is non-reactive and therefore remains 5-methylcytosine. When these reaction products are sequenced, C, which becomes U, is sequenced as a T, whereas 5-methylcytosine is sequenced as a C. This difference in sequence is what's used to determine the presence or absence of 5-methylcytosine. As far as reduced representation bisulfite sequencing goes, there's a pictorial example of how RRBS reduces the representation. Shown on the left is a bioanalyzer trace of human genomic DNA that's been digested with the restriction enzyme MSP1. You can see the bulk of the fragments are quite large. However, because MSP1 sites are highly enriched in CPG-dense regions, the resulting small fragments shown in the inset are highly enriched for CPG islands and other highly epigenetically important CPG sites. When this restriction-digested material is converted into a bisulfite-converted library for sequencing, as shown on the right-hand panel, you can see the size distribution is dramatically different. The large fragments are no longer present in the library, and what's left over is about 2% of the entire genome. This is where the dramatic reduction in sequencing costs occur. Let's get into a little more detail on the molecular biology of RRBS. At the top, I'm showing two genomic fragments, each flanked by MSP1 sites. On the left is a genomic fragment that's completely methylated at all of its CpG residues. On the right is a non-methylated fragment. The first step of the process is to digest the genomic DNA with MSP1. This restriction enzyme cuts at CCGG regardless of its methylation state. So all fragments are digested. The result are these 5' sticky end overhangs, which can be ligated to adapters, bisulfite converted, and sequenced. After bisulfite conversion, you'll notice that methyl C remains protected and remains methyl C, whereas non-methyl C is converted to U, and upon sequencing appears as a T in the sequence. One other interesting feature of RRBS, where restriction digest with MSP1 is used, is that the start of every sequence contains a DNA methylation measurement, either CGG in the case of a methylated locus, or TGG in the case of a non-methylated locus. So the information content of each sequence read is quite high. It has at least one methylation measurement, if not more. I want to describe 4 major benefits of the NuGen RRBS approach. These include direct ligation, which simplifies and speeds up the workflow. The so-called diversity adapter, which improves sequence quality without the need for Phi-X spiked in when doing Illumina sequencing. Barcode selection, which simplifies multiplexing and improves parsing of your data. And something we call N6 duplicate marking, which for the first time gives RRBS researchers the ability to identify PCR duplicates. We're going to describe each of these features in more detail. Firstly, direct ligation, as shown in this workflow diagram, allows us to go from MSP1 digestion directly to adaptor ligation. There are no steps of blend ending or A-tailing required, thus simplifying and increasing the speed of the entire process. After adaptor ligation, we perform a final repair step, bisulfite conversion, PCR amplification, and the resulting purified library has been ready for sequencing on the Illumina platform. Sequencing on the Illumina platform of RRBS libraries can be challenging. This is because the first 3 bases of the sequence contain either a TGG or a CGG, so the sequence diversity is quite low. The principle of the Illumina sequencing by synthesis chemistry is shown on this slide, where clusters are randomly generated on the surface of a flow cell. And the algorithm looks at the first few cycles to distinguish the identity of overlapping clusters. So what's shown in the top panel is a normal case of high sequence diversity library being sequenced. You can see 2 overlapping clusters have different colors, allowing the algorithm to distinguish clusters 2 from 3. However, in the case of a low diversity sample, such as an RRBS library, oftentimes you get clusters, as shown in the lower panel, where all clusters contain the same color in each cycle. This makes it very difficult to distinguish overlapping clusters. As shown in the bottom, cluster 2 is misidentified as a single cluster when in fact it's overlapping. The effect of this is that as sequencing progresses and the 2 clusters diverge in their sequence, the quality scores of that cluster become quite low because it's actually a mixed cluster. The effect of this is shown on this slide, which is a traditional RRBS library that's been sequenced on the Illumina platform. I downloaded this data from the sequence read archive, and you can see on the left-hand panel is shown the base-by-base sequence composition of the forward read. And you can see in base 1, There are both T and G signals, whereas in bases 2 and 3, the predominant signal is G, shown in black, followed by all 4 bases, with C being represented at a small fraction according to only the 5-methyl-C contained in the sample. This low diversity translates into low quality scores, as shown in the panel on the right, where we're showing the per base quality score. The yellow boxes indicate the 25 to 75% interquantile. The blue line represents the mean quality score, and the red line, which is centered around 2, represents the median quality score. So traditionally, sequencing RRBS libraries on the Illumina platform has been a challenge. The way NuGen has improved upon this method is by adding diversity, as shown in this slide. The top panel shows a representation of the reduced representation library, where an MSP fragment is shown in black in the center, flanked by the forward flow cell and forward sequencing primer sites on the left, and the reverse read barcode and reverse flow cell sites on the right. Between the forward read and the MSP1 insert, we've inserted between 0 and 3 bases of random sequence. We've done the same thing between the reverse read primer site and the MSP fragment. The result of this is demonstrated below in the 2 panels. So on the left-hand panel is a normal RRBS library, like I showed you on the previous slide. In this chart, we're showing the per base sequence composition of the sequencing run. And at base 1, you see 2 colors representing C and T. Bases 2 and 3 are essentially 100% G. That low sequence diversity is what makes sequencing so challenging. We've overcome that by adding, as I mentioned, between 0 and 3 random bases. The result is shown on the right with the NuGen RRBS with diversity. You can see all 4 bases are represented at each cycle. Therefore, improving the ability of the sequencing algorithm to identify clusters. The result is shown on this slide, where we sequenced RRBS libraries on a HiSeq without any Phi-X or any other high-diversity samples spiked in. Yet on the left, you can see the per-base sequence distribution looks like an RRBS library, T or C at the first position, followed by 2 Gs. And on the right, you can see the very high quality scores at each base of the sequencing read. So RRBS libraries produced with NuGen's system show good sequencing quality without additional Phi-X spike-in. Another feature of the system is the careful selection of barcodes. We chose barcodes with an edit distance 3, which means that you can allow one mismatch and still accurately parse your sample. The other thing we've done is we've carefully selected barcodes in what we call duplex sets, which are color balanced, meaning that it's simple to multiplex at a duplex level or higher by simply selecting any duplex set. This makes loading your sequencer in a multiplex fashion much simpler. Another feature has to do with identifying PCR duplicates. And I want to explain how traditionally PCR duplicates have been identified. This involves mapping read pairs back to the genome. The genome reference is shown in black. The read pairs are shown as arrows pointing towards each other. These read pairs have been mapped back to the genome. And then in the traditional method of identifying PCR duplicates, we look for read pairs that both start and end at the same reference genome position, as shown by those dashed red lines. So you can see 2 read pairs that share the same start and stop sites. One of them has been marked yellow because it was identified as a PCR duplicate. Those can be removed from your analysis, making your final data analysis more precise. However, you can imagine this approach does not work well for our RBS because all reads mapping to this location start and end at MSP1 site. So there's no way to tell with this traditional approach whether these sequence reads came from unique genomic molecules or in fact represent PCR duplicates. The way NuGen has approached this is to add a random 6-space tag next to the barcode, as shown in the top schematic. If you want to use this feature of duplicate marking, you simply increase your index read from 6 bases to 12 bases. The first 6 bases are the barcode, which are used for parsing and demultiplexing your sample. The next 6 bases read this N6 sequence, which allow duplicate marking, as illustrated below. Fragments are mapped back to the genome, and then all fragments that start and end at the same position are analyzed for their N6 content. As shown in this example, 2 reads share the same N6 sequence as shown in red, indicating that they came from the same adaptor ligated molecule. One of them can be marked as a PCR duplicate and removed. This approach is not available in any other RRBS method, allowing you a way to qualify that your library is of high complexity and that you're not sequencing PCR duplicates unnecessarily. Here's a demonstration of how this can be used in action. Here we've looked at 3 barcoded libraries, and on the right-hand side is an analysis that attempted to look at PCR duplicates by position only. You would never do this in an RRBS experiment because, as I showed you, all reads that map to the same genomic location start and end at the same position. And as you can see, a very high percentage of them do so. However, we can distinguish true PCR duplicates from unique genomic molecules by using this N6 duplicate marking as shown in the center panel, where we see less than 10% of the reads are actually PCR duplicates, indicating that these libraries were of high quality and high complexity. In this slide, I'm showing you 3 technical replicates of human cell line IMR90, 25 nanograms of input each. Shown in the table are uniquely mapping reads, non-uniquely mapping reads, in other words, reads that map to multiple locations in the genome, and the total of those 2. The total mapping reads are 90%, meaning that the library itself is of high quality and high mapping rate. Below are methyl CG, CHG, and CHH content. In mammalian genomes, primarily CG loci are methylated, and in this case, we see approximately 44% of those CGs are methylated. CHG and CHH are typically not methylated, and these serve as bisulfite conversion controls. We would expect very low CHG and CHH methylation. In fact, that's what we observe. On the right-hand panel is a Venn diagram demonstrating the high concordance between these 3 technical replicates. We're showing CPG loci covered at 5x depth in 1, 2, or all 3 libraries. In the ideal case, we'd like as much overlapping CPG loci identification as possible, and we see exactly that, indicating high concordance of CPG loci sequenced, and this allows you to measure differential methylation reproducibly across samples. We also want the DNA methylation levels that are measured to be reproducible and accurate, and it's shown in this concordance graph are the DNA methylation levels on the left of 2 technical RRBS replicates, and you can see the Pearson correlation is quite high. On the right-hand panel is a comparison of methylation levels measured by the RRBS approach on one axis and whole genome bisulfite on the other axis. This was data taken from a published whole genome bisulfite experiment performed on the same IMR90 cell line. And again, you see very high concordance levels. The histograms along the top and right-hand side show the distribution of data between 0 and 100% methylation. The data shows the typical bimodal distribution, where most CpG loci are either unmethylated or methylated, as expected. In these graphs, we're looking at the fraction of CpG loci covered at 5x, 10x, or 20x depth on the y-axis as a function of the number of uniquely mapping reads on the x-axis. You can use these graphs to determine how much sequencing you need to do to reach a certain level of CpG methylation depth of sequencing. On the left-hand side is one replicate of a 25 nanogram IMR90 sample. And because we ran 3 technical replicates, we were able to combine the data, as shown on the right-hand side, to demonstrate the saturation of CpG loci with increased depth of sequencing. One of the nice features of RRBS is its focus on CpG islands. In this slide, we've done some in silico analysis of the HG19 genome. We've broken it into CpG islands. As Steve mentioned, these are regions of 200 or more base pairs of the genome with a high percentage of CpG sites. CpG shores, which are the 2,000 bases flanking either side of a CpG island, promoters, and then the genome as a whole. You can see the number of regions in each class, the average size and base pairs of each region, the total number of CpGs in each class. And because after bisulfite conversion, the top strand and the bottom strand of the genome are no longer identical, they can be sequenced independently, and therefore the total number of CpGs is twice the number observed on the top strand. So we can see, for example, CpG islands, there are slightly more than 4 million CpG loci on both strands of the genome. On the bottom panel are the CpGs covered with a 100 nucleotide single-end RRBS library sequencing experiment. We did a predictive analysis where we considered MSP fragments from 40 base pairs to 300 base pairs in size, and we chose that because that was typical of early RRBS papers where a gel sizing step was used to isolate these particular size fragments. So the predicted number of CpGs in CpG islands is about 2.1 million. When we look at our actual RRBS data, we observe about 2.3 million CpGs. This is because we see MSP1 fragments slightly smaller and slightly larger than the 40 to 300 base pair size window. All told, this represents about 55% of CpGs located within CpG islands, demonstrating how effectively the RRBS approach targets sequencing to CpG islands. Likewise, for CpG shores, we're observing about 24% of all CpG loci contained in CpG shores, and about 32% of all CpGs located within promoter regions. Genome-wide, we see about 11% of all CpGs. So I hope I've demonstrated to you how RRBS is a technique that can substantially reduce sequencing costs and thereby bring more DNA methylation studies within your research budget. We've improved workflow simplicity, making a robust assay that can be completed in a single day. And as I showed you with our diversity adapter approach, we've removed the need for 5x spiking during sequencing. And in the concordance and Venn diagram data, I've demonstrated the highly reproducible data that's generated by this approach. Thanks for your attention, and we'd be happy to take some questions at this point. With that, I'll send it back to John Sterling.
Thank you, Ben. And Ben and Steve, thank you both for showing how epigenetic scientists can carry out much more effective, efficient, and economical research. Your presentations were also extremely clear, concise, and all that was very much appreciated. You've definitely struck a chord with our audience, as we've had some very interesting questions, very quality-full questions. So why don't we take the first one? All right, Ben, the first question is for you. G and C may also be methylated. How do you look at these methylations?
That's correct. So any C methylation can be detected by bisulfite conversion. G methylation is not going to be picked up with this technology.
And Ben, another question. Can this technology be applied in plants?
Yeah, this approach can be used with any genomic DNA. I would advise customers to examine the MSP digestion pattern of their particular genome before attempting the experiment. We've tested this approach in several different organisms. But as I mentioned, the relative enrichment is going to depend on the MSP1 restriction site diversity in your particular organism.
And bear with me, Ben's a long question. With RRBS, cutting with MSPL, you have a selection for high-density CPG regions like CPG islands. With this, you are missing regions with lower-density CPG regions, which are important, like enhancer regions. What's your opinion about this?
That's a great question. So with any enrichment technology, there are going to be trade-offs. In this case, we're trading off coverage of the entire genome for a much reduced sequencing cost. The RRBS technique using MSP1 is designed to target CPG-rich regions, as the question indicates. Given that we tend to observe very good coverage in things like CPG islands, promoter regions, but regions with lower MSP1 density are not covered as well. So the trade-off is much reduced sequencing cost, but also CPG poor regions of the genome are going to be underrepresented.
And then another question: do you have a preferred analysis software for working with bisulfite sequencing data?
The data I showed was analyzed using software called Bismarck that's freely available from the Babraham Institute. However, there are many different software packages available for DNA methylation analysis. Bismarck is just one of many.
And does the RRBS workflow work with degraded DNA such as from FFPE samples?
It can, with the caveat that obviously with degraded samples you're going to require higher input levels to achieve satisfactory results. One of the advantages of the RRBS workflow that we've implemented in this system is that we directly ligate adapters to the CG overhangs generated by MSP1 fragments. In other versions of the RRBS protocol where fragments are first end repaired to make them blunt and then A-tailed, any fragmentation or degraded fragment is going to be end repaired and then ligated with adapters, and it will show up in your library. The issue with that is that now your reduced representation suffers because you're now able to observe fragments that are due to degradation and not due to MSP1 digestion. With the approach that we've taken, where we directly ligate to MSP generated ends, we tend not to see those degradation products. the result of which is that we maintain the same CpG loci across all samples. So to summarize, the FFPE type material, which can be somewhat degraded, is still going to give you the same loci coverage, albeit at lower complexity, which means that you'll probably want to start with a higher input of material in such cases.
Okay, thank you. And Ben, a question. The intro slide said your minimum input for RRBS is 100 nanograms, but you show data for 25 nanograms. What's the difference?
Why don't I let Steve Kane field that question? Steve?
Yes, so when Nugen develops new kits such as the RRBS system, we tend to be conservative in the recommended minimum input amounts of material. So the 100 nanogram refers to the amount of material that we would recommend for good quality genomic DNA, and that's the minimum level of input that we use in quality control testing of our kits. So as you saw in the data section of the webinar, we have been successful of using lower input amounts as low as 25 nanograms. So in general, we would recommend that researchers first look at as high of input amount as is available based on their sample, with a minimum being 100 nanograms in sort of a pilot study phase to get a sense of how the technology is performing in their hands. And then they could certainly investigate lower input amounts as well, depending on the quality of their material and the desired results in terms of the density of CpG islands that they would detect.
Okay, thank you very much. And does the added diversity sequence affect mapping to the reference genome, Ben?
Oh, that's a great question. So we do add some extra diversity to the beginning of the read. However, that diversity is recognized and removed by a script that we provide, so that after removing that diversity, what's left is simply the MSP1 fragment sequence itself. So the answer is no, that diversity does not affect the final results.
And Ben, what is the Phi-X spike-in and how is it used?
So Phi-X is a library that Illumina sells, made out of DNA from the Phi-X 174 viral genome. It's used as a control library. It represents high quality, high sequence diversity library that can be used for a number of different controlled purposes. One purpose that Illumina recommends is for low complexity samples, such as bisulfite converted DNA or amplicon sequencing. Illumina recommends spiking in some of this Phi-X library to improve the overall cluster diversity. I described this issue of cluster diversity during my talk, and we've elected to add some diversity directly to the beginning of each read to reduce the requirement for Phi-X spike-in. I'll also mention that one of the issues with the Phi-X library is that unlike most other libraries that people sequence on the Illumina platform, the Phi-X library does not contain a barcode sequence, and that can sometimes lead to some issues while sequencing the index read. Those clusters generated from the Phi-X library tend to be dark. So the result of that is a somewhat more complicated analysis. So while we do recommend spiking in a little bit of a control library just as a way to ensure the quality of your run was good, it might be advantageous to use a library that actually contains a barcode rather than the Phi-X spike-in.
And Ben, what organisms can be used with the RRBS workflow?
So the RRBS workflow simply requires genomic DNA that's digested with MSP1. That being said, the coverage of your genome is going to vary depending on the position of those MSP1 sites. To date, We've tested this approach on human, mouse, and rat genomic DNA. The analysis in silico is fairly straightforward. You can simply analyze your genome for the MSP1 sites and then determine essentially coverage of your genome by selecting MSP1 fragments that are approximately 30 bases to 300 base pairs in length. Those tend to be the fragments that are going to be analyzed by this RRBS technique.
Ben, how do you avoid the gel size selection step in the workflow?
Yeah, so early versions of the RRBS method were published in which there was a size selection step by gel electrophoresis. Later versions eliminated that step, thankfully, because it is a rather tedious and time-consuming step. Essentially, at the low end of fragment size, those fragments are removed by the purification. In our case, we have a bead-based purification approach. So very small fragments are removed that way. On the large size end of the spectrum, we have a bisulfite conversion step, which tends to degrade DNA to some extent. The larger the fragment, the more likely that fragment is going to contain some sort of a hydrolysis event. And once that happens, that fragment is no longer represented in the library. So as a basically natural course of the bisulfite conversion process, we see a trailing off of fragments much above 500 base pairs. And more typically, we see mostly fragments less than 300 base pairs. So that combination of purification and bisulfite conversion allow us to complete the whole library construction process without a gel sizing step.
Ben, thank you very much for that detailed answer. Unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. I want to say thanks again to the panel for their outstanding presentations, and thank you to our audience for your attention and for your very thoughtful questions about the various topics brought up during the webinar. And thank you to Nugent Technologies, whose support made this webinar possible. Bye for now.